So here's something worth thinking about as you settle in tonight. Right now, without you doing a single conscious thing, your body is performing roughly 37,000 billion chemical reactions every single second. Your heart is beating, somewhere around 60 or 70 times a minute, pushing about 5 liters of blood through a network of vessels so long that if you laid them end to end, they'd wrap around the earth more than twice. Your lungs are pulling in air, extracting oxygen molecules, and passing them into your bloodstream, where they hitch a ride on red blood cells that are hurtling through your arteries at speeds that would surprise you. Your immune system is quietly patrolling, scanning for invaders, remembering threats it encountered years ago. Your stomach is digesting, your liver is filtering, your kidneys are balancing the chemistry of your blood with a precision that would make a laboratory envious. And your brain, this 3-pound mass of tissue sitting behind your forehead, is coordinating all of it while simultaneously generating the consciousness that lets you lie here and wonder about it. You don't have to think about any of this. It just happens. And for most of human history, nobody had the faintest idea how any of it worked. The human body was the most intimate mystery in the world, the one thing every person on Earth carried around with them every single day, and yet the one thing that remained almost completely opaque for thousands and thousands of years. And that's what we're going to talk about tonight. The human body, the whole astonishing, improbable, beautifully engineered thing. We're going to walk through it slowly, starting from the very beginning, from the first people who tried to figure out what was going on inside us. And we're going to take our time with it. There's no rush. If you drift off somewhere along the way, that's perfectly fine. Your body will keep running all of its systems without your conscious involvement. It's been doing that your whole life. And honestly, some of this stuff is so wonderfully intricate that it settles into you better when you're relaxed, when your mind is loose and open, and not trying to force everything into neat categories. Because here's the thing about the human body. It resists neat categories. Every system depends on every other system. The heart needs the lungs need the blood needs the kidneys need the nervous system needs the heart. It's all one interconnected, endlessly cycling, self-repairing, self-regulating miracle. And the story of how we came to understand it, even partially, is one of the great intellectual adventures in human history. It involves rivalries, accidents, moments of breathtaking insight, and centuries of being completely, confidently, spectacularly wrong. Let's start where most of these stories start, which is the ancient world. And specifically, let's start with the Greeks, because the Greeks were the first people we know of who tried to understand the body not through magic or divine will, but through observation and reason. Before them, illness was largely understood as punishment from the gods, or the work of evil spirits, or the result of cosmic imbalances that had nothing to do with what was actually happening inside a person's chest or belly. If you got sick in ancient Mesopotamia, a priest might examine the entrails of a sacrificed animal to diagnose your condition. If you got sick in ancient Egypt, a healer might recite spells from a papyrus scroll. Though, to be fair to the Egyptians, their medical knowledge was actually more advanced than they often get credit for. The Edwin Smith Papyrus, dating from around 1600 BCE, but probably based on texts 1,000 years older, contains remarkably clear-headed descriptions of surgical cases, head wounds, spinal injuries, broken bones, with observations about symptoms, prognoses, and treatments that are recognizably clinical. The Egyptians knew how to set broken bones. They understood that injuries to one side of the head could cause symptoms on the opposite side of the body, and they used honey as a wound dressing, which modern medicine has confirmed has genuine antibacterial properties. But they also mixed their practical knowledge with incantations and amulets, because they didn't have a framework for separating what worked from what didn't, or for understanding why anything worked at all. The person who changed that, more than anyone else, was Hippocrates of Kos. Born around 460 BCE on a small Greek island in the Aegean Sea, Hippocrates is often called the father of medicine, and while that title oversimplifies things, he almost certainly didn't write all the texts attributed to him, and he was part of a broader intellectual tradition. The core idea associated with his name really was revolutionary, and the idea was this: disease has natural causes, not supernatural ones, not divine ones, natural ones. The body gets sick because something has gone wrong inside it, something physical, something you can in principle observe and understand, and maybe even fix. That might sound obvious to you now, but in the 5th century BCE, it was a radical act of intellectual courage. Hippocrates and his followers developed a theory of the body built around 4 substances they called humors: blood, phlegm, yellow bile, and black bile. Health, they believed, was a state of balance among these 4 humors. When they were in harmony, you felt well. When one was out of proportion, too much blood, not enough phlegm, an excess of black bile, that's when you got sick. And treatment was about restoring the balance. Too much blood, you'd be bled, either by cutting a vein or applying leeches. Too much phlegm, you might be given something to make you vomit or sweat. It was a complete system, elegant, internally consistent, and almost entirely wrong. But here's the thing, and this matters. It was wrong in its specifics, but it was right in its approach. The Hippocratic physicians were doing something genuinely new. They were observing patients, recording symptoms, tracking the course of diseases over time, and trying to find patterns. The Hippocratic texts include case studies that are recognizable as clinical medicine. They describe fevers, infections, broken bones, epilepsy, and they describe them in terms of what the physician actually saw and heard and felt, not in terms of which god was angry. That observational tradition, that commitment to looking at the body and trying to understand it on its own terms, is the foundation of everything that came after. Now, the most influential physician in the ancient world, after Hippocrates, was a man named Galen. And Galen is one of those figures who casts such a long shadow over history that it's hard to know whether to admire him or be exasperated by him. Born around 101 CE in Pergamon, in what is now Western Turkey, Galen was brilliant, prolific, and spectacularly confident. He trained at the best medical schools of his era, including Alexandria, where the tradition of anatomical study was strongest. He served as physician to the gladiators in Pergamon, which gave him extraordinary practical experience with trauma, wounds, and the insides of living bodies, and eventually became personal physician to several Roman emperors. Galen wrote prolifically, hundreds of texts covering anatomy, physiology, pharmacology, and philosophy, and his influence was absolutely enormous. For more than 1,000 years after his death, Galen's writings were treated as essentially infallible throughout Europe and the Islamic world. If Galen said the liver had 5 lobes, then the liver had 5 lobes. If Galen said blood was produced in the liver and consumed by the organs like fuel, then that's how it worked. His authority was so total, so unquestioned, that it actually held back medical understanding for centuries. And here's the problem. Galen never dissected a human body. Roman law prohibited it, so he dissected animals instead: Barbary macaques, pigs, goats, oxen, and extrapolated from what he found to how the human body must work. And a lot of his extrapolations were wrong. The human liver doesn't have 5 lobes. That's a pig liver. The human jaw is 1 bone, not 2. That's a dog jaw. Galen described a network of blood vessels at the base of the brain, called the rete mirabile, the wonderful net, that exists in ungulates but not in humans. He believed that blood passed from the right side of the heart to the left through invisible pores in the septum, the wall between the heart's chambers. Those pores don't exist, but for 1,400 years, nobody dared to say so, because challenging Galen was almost like challenging scripture. Think about that for a second. 1,400 years. That's how long the wrong model persisted, not because it was tested and confirmed, but because the authority of the person who proposed it was so overwhelming that testing it felt like heresy. And this is a pattern you see throughout the history of medicine, the tension between authority and observation, between what the books say and what the body actually shows you when you open it up and look. And the idea of opening it up, that's where the story gets complicated and honestly, a little grim. Because for most of Western history, cutting open a human body was forbidden, or at least deeply restricted. The Catholic Church didn't explicitly ban dissection. That's a common misconception. But cultural and legal taboos made it extremely difficult. When dissection was permitted, usually for a few weeks each winter in the late medieval period, the bodies came from executed criminals, and the whole event was a public spectacle, as much theater as science. Students sat in tiered galleries, watching from a distance, while a low-ranking barber surgeon did the actual cutting, and a professor lectured from Galen's texts above. The professor often didn't even look at the body. He didn't need to. The answers were already in the book. But by the 15th and 16th centuries, something was shifting. The Renaissance was bringing a new spirit of inquiry, a willingness to look at the world with fresh eyes, rather than through the lens of ancient authority. Artists like Leonardo da Vinci were dissecting human corpses, illegally in some cases, and producing anatomical drawings of extraordinary accuracy and beauty. Leonardo's anatomical notebooks, which weren't published until centuries after his death, contain observations about the heart's valves, the structure of the fetus in the womb, and the curvature of the spine that are startlingly correct. He was drawing what he saw, not what Galen said he should see. But Leonardo was an artist, not a physician, and his work didn't enter the medical mainstream. The person who brought observation into the heart of medical education was a young man from Brussels named Andreas Vesalius, and his story is one of the great turning points in the history of science. Born in Brussels in 1514 to a family with a long medical tradition, his father was a pharmacist to the Emperor Charles V, and his grandfather and great-grandfather had also been physicians. Vesalius was drawn to anatomy almost from childhood. There are stories, possibly embellished, of the young Vesalius dissecting mice and stray cats and dogs in his spare time, which is either charming or disturbing depending on your temperament. He studied at the University of Paris, where anatomy lectures were still conducted in the medieval style. A professor sat in an elevated chair, reading aloud from Galen, while a barber surgeon did the actual cutting below, and students watched from a distance, learning anatomy from the book rather than the body. Vesalius found this infuriating. He wanted to do the cutting himself. He wanted to get his hands inside the body and see what was actually there, not what Galen said should be there. And getting his hands on bodies wasn't easy. The supply of cadavers for dissection was extremely limited, a few executed criminals per year, if you were lucky, and students like Vesalius had to get creative. There are accounts of Vesalius sneaking out to the Cemetery of the Innocents in Paris to collect bones, or retrieving the body of an executed criminal from a gibbet outside the city walls and smuggling it back to his rooms piece by piece. This wasn't unusual for the time. The demand for anatomical specimens always exceeded the legal supply, and ambitious anatomists routinely resorted to grave robbing, bribery of executioners, and other methods that were, to put it politely, legally questionable. The body-snatching trade would become a full-fledged industry in the 18th and 19th centuries, with professional resurrection men digging up freshly buried corpses and selling them to medical schools. But that's a story for another night. And when he did, he started finding things that didn't match. The human jaw was one bone, not two. The sternum had 3 segments, not 7. The vena cava originated from the heart, not the liver. Each discrepancy was a crack in Galen's edifice, and Vesalius, to his enormous credit, didn't look away. He documented them, meticulously. In 1543, and that's a very big year in the history of science, because it's also the year Copernicus published his theory that the Earth orbits the Sun, Vesalius published a book that would revolutionize anatomy. It was called De Humani Corporis Fabrica, which translates roughly as On the Fabric of the Human Body, and it was breathtaking. 7 volumes, over 700 pages, filled with detailed, anatomically accurate illustrations that were light years beyond anything that had come before. The illustrations, probably produced by artists from the studio of Titian in Venice, showed human figures in dynamic poses, standing, gesturing, seemingly alive, with their skin peeled back and their muscles and organs exposed. They were beautiful and eerie and scientifically meticulous, and they showed the human body as it actually was, not as Galen had imagined it to be. The reaction was fierce. Some of the medical establishment embraced Vesalius. Here, finally, was real anatomy, based on real human dissection, correcting errors that had persisted for over a millennium. But others were outraged. Vesalius's own teacher, Jacobus Silvius, attacked him bitterly, calling him a madman and suggesting that if the human body didn't match Galen's descriptions, then the body must have changed since Galen's time, not that Galen had been wrong, which is an almost comical level of intellectual contortion. But it shows you how powerful the hold of authority was. Silvius would rather believe that human anatomy had physically evolved in 1400 years than accept that a revered ancient author had made mistakes. Vesalius himself had a complicated life after the Fabrica. He became court physician to Emperor Charles V and later to Philip II of Spain, largely abandoning his anatomical research. In 1564, he embarked on a pilgrimage to the Holy Land. The reasons are debated. Some say it was penance for a dissection mishap. Others say it was political, and died on the return journey, on the Greek island of Zakynthos, at just 49 years old. But the revolution he started was irreversible. After Vesalius, the study of anatomy was built on observation, on dissection, on looking at the actual body. The age of uncritical deference to ancient texts was over, and the discoveries that flowed from this new approach came fast. One of the most important came from a quiet, methodical Englishman named William Harvey, and it had to do with something so fundamental, so basic to your existence, that it's beating inside you right now, your heart. And to understand why Harvey's discovery was so revolutionary, you need to understand what people believed about the heart and blood before he came along, because it was, and I say this with affection for the history of medicine, a spectacular mess. The prevailing model, still basically Galenic, went something like this. Food gets digested in the stomach and intestines, turned into a substance called chyle, which travels to the liver. The liver transforms chyle into blood and imbues it with something called natural spirit. This blood then flows outward through the veins to the organs, which consume it as fuel, like wood burning in a fire. Some blood reaches the right side of the heart, where it passes through those invisible pores in the septum to the left side, mixing with air from the lungs to create vital spirit. This vitalized blood then flows through the arteries to the body, carrying life force. Blood, in this model, is constantly being produced in the liver and constantly being consumed by the body. It doesn't circulate. It's manufactured and burned, like a one-way fuel system. Harvey didn't buy it. Born in 1578 in Folkestone, on the English coast, he studied at Cambridge and then at the University of Padua in Italy, which had become the leading center of anatomical research in Europe after Vesalius. At Padua, Harvey studied under a professor named Hieronymus Fabricius, who had discovered the valves in veins, little flaps of tissue inside the veins that allowed blood to flow in one direction, but not the other. Fabricius didn't know what the valves were for. He thought they might slow the flow of blood to prevent the extremities from getting too much. Harvey looked at those valves and started thinking. Here's what Harvey did that was so clever. He started with a simple quantitative argument, something almost nobody in medicine had done before. He estimated the amount of blood the heart pumps with each beat. The left ventricle, he calculated, holds about 2 ounces of blood. If the heart beats 72 times a minute, that's roughly 144 ounces per minute. Over an hour, that's more than 540 pounds of blood. In a day, more than 6.5 tons. Now, think about that. If blood is being constantly manufactured in the liver and consumed by the body, as Galen taught, then the liver would need to produce 6.5 tons of blood from food every single day. That's obviously impossible. The quantity alone demolished the old model. Harvey published his findings in 1628 in a thin book called Exercitatio Anatomica de Motu Cordis et Sanguinis in Animalibus, an anatomical exercise on the motion of the heart and blood in animals, usually just called de motu cordis. And what he described was circulation. The heart is a pump. It pumps blood out through the arteries to the body. The blood returns through the veins back to the heart. The valves in the veins ensure one-way flow, back toward the heart, which is why Fabricius had found them there. The blood isn't consumed. It goes around and comes back. It circulates. It's one continuous loop. This was the first major physiological discovery, based on experimental evidence and quantitative reasoning. Harvey didn't just observe. He measured, he calculated, he experimented. He tied tourniquets around arms and watched how veins swelled on one side and emptied on the other, the side away from the heart, which proved blood in veins flows toward the heart, not away from it. He examined the hearts of more than 80 species of animals. He watched the hearts of cold-blooded animals, which beat slowly enough that you can actually see the sequence of contraction, the atria squeezing first, then the ventricles. He even observed the beating heart of a shrimp. He was systematic, rigorous, and patient. And he was right. The reaction to Harvey's work was mixed, which might be the polite way of putting it. Many physicians rejected his ideas outright. Some accused him of heresy. His medical practice suffered. People didn't want to be treated by a doctor with dangerous new ideas. But Harvey was patient and careful, and over the following decades, his evidence proved so compelling that opposition gradually crumbled. By the time he died in 1657, the circulation of the blood was on its way to becoming accepted medical fact. But here's the thing. Harvey couldn't explain quite how blood got from the arteries to the veins. He knew it happened. His logic demanded it. But the vessels connecting arteries to veins were too small to see with the naked eye. He predicted their existence, but he couldn't prove it visually. That proof would come 30 years after Harvey's death, when an Italian anatomist named Marcello Malpighi pointed a microscope at the lung of a frog in 1661 and saw them, tiny hair-like vessels connecting the smallest arteries to the smallest veins. He called them capillaries, from the Latin capillaris, meaning of or relating to hair. And there they were, the missing piece of Harvey's circuit. The body's tiniest blood vessels, so small that red blood cells have to squeeze through them in single file, and so numerous that every living cell in your body is within a few cell widths of a capillary. That's how the delivery system works. Oxygen and nutrients pass through the thin capillary walls into the tissues, and waste products pass back the other way. It's elegant, it's efficient, and it's happening everywhere in your body right now, in billions of capillaries simultaneously. And your heart. Let's stay with your heart for a moment, because it deserves it. Think about what this organ actually does. It's about the size of your fist, sitting slightly left of center in your chest, between your lungs. It's mostly muscle, cardiac muscle, a type of muscle that exists nowhere else in your body, muscle that never gets tired the way your biceps do, muscle that contracts and relaxes, about 100,000 times a day, roughly 2.5 billion times in an average lifetime. And it does this without you ever telling it to. You don't decide to beat your heart. It beats on its own. The way it does this is genuinely beautiful. Your heart has its own built-in electrical system, a cluster of specialized cells called the sinoatrial node, or SA node, sitting in the wall of the right atrium. These cells spontaneously generate electrical impulses about 60 to 100 times a minute. No nerve signal from the brain tells them to fire. They just do. Each impulse spreads across both atria, the heart's upper chambers, causing them to contract and push blood down into the ventricles, the larger lower chambers. Then the signal reaches another cluster, called the atrioventricular node, which delays it for about 1/10 of a second, just long enough for the ventricles to fill completely, and then passes it along specialized conducting fibers that spread the impulse rapidly across both ventricles, causing them to contract in a powerful, coordinated squeeze that pushes blood out into the arteries. The right ventricle sends blood to the lungs to pick up oxygen. The left ventricle sends blood to the entire rest of the body. And then the whole cycle starts over. And the engineering of those valves, 4 of them in total, the mitral and tricuspid valves between the atria and ventricles, and the aortic and pulmonary valves at the exits, is exquisite. They're passive devices, opening and closing based purely on pressure differences. No muscles pull them open or shut. Blood pressure does all the work. When the ventricle pressure exceeds the arterial pressure, the exit valve opens and blood flows out. When the ventricle relaxes and the pressure drops, the valve snaps shut, preventing backflow. And those valves open and close 100,000 times a day, every day, for decades, without wearing out. When they do eventually fail, when a valve narrows or leaks, it's often after 70 or 80 years of continuous operation, which is an engineering record that no human-made valve has come close to matching. That tiny delay at the AV node, that 1/10 of a second, is the reason your heartbeat has that characteristic lub-dub sound. The first sound is the atria contracting, and the valves between the atria and ventricles snapping shut. The second sound is the ventricles contracting, and the valves at the exits of the ventricles closing. 2 sounds, 1 coordinated sequence, 100,000 times a day. And the network of vessels this blood travels through. Let's appreciate that for a moment. Your body contains roughly 60,000 miles of blood vessels. The aorta, the largest artery, is about the diameter of a garden hose and carries oxygenated blood away from the left ventricle at considerable pressure. From the aorta, arteries branch and branch again, getting progressively smaller. Arteries to arterioles to capillaries, like a tree trunk splitting into branches, then twigs, then the finest filaments. The walls of the arteries are thick and muscular, designed to withstand the pressure of each heartbeat. The walls of the capillaries are just 1 cell thick, the thinnest walls in your entire body, because their job requires it. Nutrients and oxygen need to pass through those walls into the tissues, and waste products need to pass back. And then the capillaries merge into venules, which merge into veins. which carry the deoxygenated blood back to the heart at much lower pressure. Veins have thinner walls than arteries because they don't need to withstand the same forces. And they have those valves Harvey was so interested in, the one-way flaps that prevent blood from pooling in your legs under the pull of gravity. And the blood traveling through all of this, that's worth talking about too, because blood is far more complex and interesting than most people realize. It's not just red liquid, it's a living tissue, the only tissue in your body that's liquid, and it contains an entire ecosystem of specialized cells, each with its own job. Red blood cells, about 25 trillion of them in your body right now, are the oxygen carriers. They're packed with hemoglobin, a protein built around an iron atom, and it's the iron that gives blood its red color and that bonds with oxygen molecules in the lungs and releases them in the tissues. Each red blood cell lives about 120 days, and your body produces roughly 2 million new ones every second to replace the ones that are dying. 2 million every second. Your bone marrow is a factory that never shuts down. White blood cells are the defense force. We'll talk about them more later, because the immune system is its own incredible story. Platelets are the repair crew, tiny cell fragments that rush to the site of any injury and clump together to form clots, sealing wounds and stopping bleeding. And all of these cells are floating in plasma, a yellowish liquid that makes up about 55% of your blood's volume and carries dissolved proteins, hormones, nutrients, waste products, and antibodies. Your blood is a river of information and resources, carrying messages from one part of the body to another, delivering fuel, removing waste, defending territory. It's your body's logistics network, its communication system, and its military, all rolled into one. But let's zoom out from the heart and blood for a moment, because the discovery that really cracked open the deepest secrets of the human body, the discovery that revealed a whole universe hidden inside us, invisible to the naked eye, that came not from a physician or an anatomist, but from a Dutch draper who ground lenses as a hobby. His name was Antony van Leeuwenhoek, and his story is one of the most delightful and unlikely in the entire history of science. Leeuwenhoek was born in 1632 in Delft, in the Netherlands. He had no university education. He wasn't a scientist in any formal sense. He was a textile merchant, a draper, and he started grinding lenses because he wanted to inspect the quality of cloth more closely. He needed magnification to count the thread density in fabrics. But his lenses were extraordinary. Through painstaking craftsmanship, he never shared his exact methods, and some of his techniques are still debated. He produced tiny, single-lens microscopes that could magnify objects up to nearly 300 times. That doesn't sound like much compared to modern microscopes, but for the 17th century, it was astonishing. Nobody else could make lenses this good. And then Leeuwenhoek started looking at everything. He looked at pond water and saw tiny creatures swimming around. He called them animalcules, little animals. He was seeing bacteria and protozoa, organisms that no human eye had ever observed before. He looked at a scraping from his own teeth and saw more of these tiny organisms living in the plaque between his molars. He examined blood and saw the red blood cells, describing them as flat, oval, and remarkably uniform. He looked at muscle fibers, at the structure of hair, at the crystalline patterns in minerals, at the spermatozoa in semen. He was the first person to observe and describe them, which caused some awkwardness when he reported his findings to the Royal Society in London. Leeuwenhoek was particularly fascinated by the creatures he found in water. He collected samples from rain barrels, from lakes, from his own mouth, from the feces of animals, and he recorded what he saw with meticulous detail, describing sizes relative to grains of sand, recording their movements, their shapes, their behavior. He saw rod-shaped bacteria, spherical bacteria, spiral-shaped bacteria, organisms that would later be classified as the major morphological types of the bacterial kingdom. He was, in a sense, the first microbiologist, and he was doing it with a lens he held up to his eye, like a jeweler inspecting a gemstone. Because that's the thing. Leeuwenhoek sent his observations to the Royal Society in London, written in Dutch, describing things that nobody had ever seen before, from a man with no credentials and no formal training. The Fellows of the Royal Society were skeptical at first, as you'd expect, but they sent a delegation to Delft to verify his claims, and when they looked through his microscopes, they saw exactly what he described. The tiny creatures were real. The invisible world was real. And Leeuwenhoek spent the next 50 years of his life exploring it, writing over 560 letters to the Royal Society, each one describing some new marvel he'd found swimming or crawling or floating in a drop of water, a fleck of dirt, a sample of blood. Now, around the same time, actually slightly earlier, an Englishman named Robert Hooke was doing his own microscopy work. In 1665, Hooke published a book called Micrographia, filled with gorgeous illustrations of things he'd seen under the microscope: the eye of a fly, the barb of a stinging nettle, the structure of a feather. And in one of the most famous observations in the history of biology, Hooke examined a thin slice of cork under his microscope and saw that it was made up of tiny, repeating, box-like structures. They reminded him of the small rooms, cells, that monks lived in at a monastery, so he called them cells, and that name stuck. Now, Hooke was looking at dead cork, so what he actually saw were the cell walls of dead plant cells. The cells themselves were long gone. It would take nearly 200 years for scientists to piece together the full picture. In the 1830s, a German botanist named Matthias Schleiden proposed that all plants are made of cells. Shortly after, another German scientist, Theodor Schwann, extended the idea to animals, and in 1855, Rudolf Virchow added the crucial third piece: all cells come from other cells. Omnis cellula a cellula. Cells don't spontaneously generate from non-living matter. Every cell in your body can trace its lineage back, cell division by cell division, in an unbroken chain stretching back billions of years to the very first living cell on Earth. You are, quite literally, the product of an uninterrupted line of cell division that has never stopped since life began. Think about that. The cells in your body are the direct descendants, daughter cells of daughter cells of daughter cells, billions of generations deep, of cells that were dividing when the Earth was young and the oceans were new. Not a single generation was skipped. Not a single division failed fatally. An unbroken thread of living continuity, stretching from you to the origin of life. And the cells themselves, let's appreciate what a single cell actually is, because it's easy to treat cells as just the building blocks of the body, the way bricks are the building blocks of a house. But a cell is not a brick. A cell is itself a fantastically complex system. A typical human cell contains thousands of different types of proteins, each with a specific job. It has mitochondria, organelles that generate the cell's energy by converting glucose and oxygen into ATP, the molecular fuel that powers virtually everything the cell does. Your mitochondria produce roughly your own body weight in ATP every single day, and then recycle it over and over. The cell has ribosomes, tiny molecular machines that read genetic instructions and assemble proteins, amino acid by amino acid. It has an endoplasmic reticulum where proteins are folded and modified, and a Golgi apparatus that packages them for delivery. It has lysosomes that break down waste and worn-out components. It has a cytoskeleton, a network of protein filaments that gives the cell its shape and allows it to move and divide. And at the center, protected by a double membrane, sits the nucleus, containing the DNA, the master blueprint, coiled and compacted into chromosomes. All of this, in a cell about 1/10 the width of a human hair. And cell theory, the idea that all living things are made of cells, and that all cells come from pre-existing cells, this wasn't just a nice biological fact. It was the key that eventually unlocked one of the most important medical advances in human history, the germ theory of disease. Because once you know that tiny living organisms exist, organisms invisible to the naked eye, the question becomes, could those organisms be causing disease? For most of history, the dominant theory of disease transmission was miasma, bad air. People believed that diseases like cholera, plague, and malaria, the word malaria literally comes from the Italian for bad air, were caused by foul-smelling vapors rising from swamps, rotting matter, and filth. And in a roundabout way, they weren't entirely wrong about the correlation. Places with bad smells often were places with disease. But the smell wasn't causing the illness. The underlying conditions that produced the smell were also breeding grounds for the actual culprits, microorganisms. The person who first demonstrated this connection in the most dramatic possible way wasn't a famous scientist. He was a Hungarian obstetrician named Ignaz Semmelweis, and his story is one of the most heartbreaking in all of medicine. In the 1840s, Semmelweis was working at the Vienna General Hospital, where they had 2 maternity wards. One was staffed by doctors and medical students. The other was staffed by midwives, and the maternal death rate in the doctor's ward was horrifying, sometimes 5 times higher than in the midwives' ward. Women begged not to be admitted to the doctor's ward. Some preferred to give birth on the street rather than enter it. The cause of death was usually puerperal fever, childbed fever, a devastating infection that killed new mothers in agony within days of delivery. Semmelweis was obsessed with understanding why. He ran through every possible explanation he could think of. Was it the position the women gave birth in? No. Both wards used the same positions. Was it the food? The ventilation? The laundry? The type of bedding? He eliminated possibility after possibility. And then, in 1847, a clue arrived in the form of a tragedy. His friend and colleague Jakub Kolečka was accidentally cut with a scalpel during an autopsy, and within days, Kolečka developed symptoms identical to puerperal fever and died. Semmelweis was devastated by the loss, but even in his grief, the scientist in him saw the connection. Kolečka had been infected by something from the cadaver, the same kind of cadaverous material that the medical students in his ward were carrying on their hands when they went directly from the autopsy room to the maternity ward. Think about what that morning must have been like for the women in that ward. A student who had just been elbow-deep in a decaying corpse would walk in, wipe his hands on his coat, maybe rinse them quickly, maybe not, and proceed to examine a woman in labor. The stench of the cadaver was often still on his fingers. Nobody thought this was a problem. The idea that invisible substances on a doctor's hands could cause disease was, in the 1840s, simply not part of the medical worldview. Disease was caused by miasma, by bad air, by imbalances in the humors, not by something so mundane as dirty hands. Semmelweis instituted a simple policy. Everyone had to wash their hands in a solution of chlorinated lime before examining patients. The death rate plummeted, from over 10% to under 2%. In some months, to 0. It was one of the most clear-cut, dramatic demonstrations of cause and effect in the history of medicine. And the medical establishment's response was to ignore him, mock him, and eventually drive him out of Vienna. The idea that doctors themselves were killing their patients, that the hands of a gentleman physician could carry death, was intolerable to the medical profession of the mid-19th century. Semmelweis grew increasingly bitter and erratic, writing furious open letters accusing his critics of murder. In 1865, he was committed to a mental asylum, where he was beaten by guards and died 2 weeks later, possibly from an infection in a wound sustained during the beating. He was 47. The validation of Semmelweis's ideas came, ironically, from the very science that would have given him the theoretical framework he lacked. Because while Semmelweis knew that something on doctors' hands was causing infections, he didn't know what that something was. He couldn't see it. He didn't have germ theory. That came from two giants of 19th-century science who were working, partly independently and partly in rivalry, to prove that microscopic organisms cause disease. The validation came slowly, piece by piece, from some of the most brilliant experimental scientists who ever lived. Louis Pasteur, a French chemist, not a physician, a chemist, demolished the ancient idea of spontaneous generation through a series of elegant experiments in the 1850s and 1860s. He showed that microorganisms in the air were responsible for fermentation and spoilage, that broth sealed from airborne contamination stayed sterile, and that heating liquids could kill the microbes in them, a process we still call pasteurization. And then he extended these findings to disease, demonstrating that specific microorganisms caused specific diseases. Anthrax in cattle, cholera in chickens, silkworm blight, each one caused by a particular germ that could be isolated, cultured, and studied. Robert Koch, a German physician, took this further with a rigor and precision that set the standard for microbiology. Koch was a meticulous experimenter, working initially in a small laboratory in his own home, using equipment his wife had bought him as a birthday present. He developed techniques for growing bacteria in pure cultures on solid media, for staining them with aniline dyes so they could be seen clearly under microscopes, and for photographing them through the microscope to create permanent records. And most importantly, he established a set of criteria, Koch's postulates, for proving that a specific microorganism causes a specific disease. The organism must be found in all cases of the disease. It must be isolated from a diseased organism and grown in pure culture. The cultured organism must cause the disease when introduced into a healthy organism, and it must be re-isolated from that newly diseased organism. Koch used these postulates to prove that Mycobacterium tuberculosis causes tuberculosis, and that Vibrio cholerae causes cholera. These were among the most important medical discoveries of the 19th century, and they earned Koch the Nobel Prize in 1905. And from germ theory flowed modern surgery. Joseph Lister, a British surgeon who read Pasteur's work, realized that the horrifying death rates from post-surgical infections, gangrene, sepsis, wound rot, might be caused by the same kind of microorganisms Pasteur had discovered. In the 1860s, Lister began using carbolic acid to sterilize surgical instruments, clean wounds, and even spray the air in operating rooms. The results were dramatic. In his ward at the Glasgow Royal Infirmary, the death rate from post-amputation infections dropped from nearly 50% to about 15%. That's an extraordinary number. Think about what surgery was like before Lister. The operating theater, and it was literally called a theater because crowds of students watched, was a nightmare by modern standards. Surgeons wore their street clothes. They used unsterilized instruments. They might wipe a scalpel on their apron between patients. The wooden operating tables were soaked with the blood and fluids of hundreds of previous patients. The faster a surgeon could work, the better, not just to minimize the patient's pain. This was before reliable anesthesia for most of the pre-Lister period, but to minimize their exposure to infection. The famous Scottish surgeon Robert Liston could amputate a leg in under 2.5 minutes. Speed was literally a survival factor. Lister changed all of that, and while his methods were initially resisted, many surgeons found the carbolic acid spray irritating and considered the whole antiseptic protocol an unnecessary fuss. The results were impossible to ignore. Before Lister, surgery was a last resort, almost as likely to kill you as save you. After Lister, it became a viable medical intervention. Modern antiseptic and later aseptic surgical technique, sterile operating rooms, sterile instruments, surgeons scrubbing in, wearing gloves and gowns and masks, all of it flows from Lister, who built on Pasteur, who built on Koch, who built on a foundation of microbiology that Leeuwenhoek, the draper from Delft, had unknowingly laid 2 centuries earlier by peering at drops of water through his handmade lenses. But let's shift our attention now to something that connects every system we've talked about so far, something that coordinates, controls, and makes sense of everything else your body does, your nervous system, and specifically, your brain. Because if the heart is the body's engine, and the blood is its highway system, and the cells are its building blocks, then the nervous system is its internet, the communication network that links everything together and lets the body respond to the world in real time. The story of how we came to understand the nervous system begins, appropriately enough, with a dead frog and a flash of electricity. In 1780, an Italian physician and physicist named Luigi Galvani was dissecting frogs at the University of Bologna. According to the most famous version of the story, and like many famous stories, it might be embellished, Galvani's wife Lucia noticed that a frog's leg twitched when a metal scalpel touched its nerve at the same moment that a spark flew from a nearby electrostatic machine. Galvani investigated further, eventually discovering that touching the nerve with 2 different metals, say copper and zinc, also produced the twitch, even without the static machine. The frog's legs could be made to kick by purely electrical stimulation. Galvani was electrified, if you'll pardon the expression. He spent years investigating this phenomenon, eventually concluding that animal tissues contain an innate form of electricity, animal electricity, he called it, distinct from the electricity in lightning or static machines. He was partly right and partly wrong. His rival, Alessandro Volta, argued that the electricity was coming from the contact between the 2 different metals, not from the animal tissue itself, and Volta went on to invent the voltaic pile, the first true battery, to prove his point. The rivalry was bitter and lasted years, but Galvani's fundamental insight that electrical signals play a role in how the body works, that nerves carry something like an electrical current, that was correct, and it opened a door that would take another century and a half to walk through fully. In the decades that followed, other researchers expanded on the connection between electricity and the nervous system. In the 1840s, the German physiologist Émile Dubois-Reymond demonstrated that nerve impulses are accompanied by measurable electrical changes, and his colleague Hermann von Helmholtz actually measured the speed of nerve impulses in a frog, finding them much slower than the speed of electricity in a wire, only about 25 to 30 meters per second. This was an important clue. If nerves conducted electricity the way wires do, the signal should travel at the speed of light. The fact that it traveled much more slowly suggested something fundamentally different was going on, something more biological, more complex than simple electrical conduction. What we now know is this: your nervous system is made up of specialized cells called neurons, and you have roughly 86 billion of them in your brain alone, with trillions more spread throughout your spinal cord and peripheral nerves. A neuron is an extraordinary cell. It has a cell body, like any other cell, but it also has long, thin extensions. A single long one, called an axon, which can be astonishingly long, up to a meter in some cases, running from your spinal cord all the way down to your foot. and shorter, branching ones called dendrites. The dendrites receive signals from other neurons. The axon transmits signals to other neurons, muscles or glands. And the way the signal travels down the axon is genuinely clever, and the story of how we figured it out involves one of the great debates in neuroscience history. In the late 19th century, there was a fierce argument about whether the nervous system was a continuous network, one unbroken web of tissue, or whether it was made up of individual, separate cells. An Italian physician named Camillo Golgi developed a staining technique using silver that made individual neurons visible under the microscope for the first time, revealing their beautiful, branching shapes in startling detail. But Golgi believed the neurons were fused together into a continuous mesh. A Spanish anatomist named Santiago Ramón y Cajal used Golgi's own staining technique to produce extraordinary drawings of neurons and argued passionately that they were separate cells communicating across tiny gaps. Cajal was right, and both men shared the Nobel Prize in 1906, which must have made for an awkward ceremony. But the actual mechanism of neural signaling, the way the signal moves, is not like electricity flowing through a wire. It's more like a wave, a domino effect. The axon's membrane has tiny channels that can open and close, letting charged particles, ions of sodium and potassium, flow in and out. When the neuron fires, sodium channels open at one spot, letting positively charged sodium ions rush in. This depolarization triggers the next set of channels to open, and the next, sending a wave of electrical change racing down the axon at speeds ranging from about 2 meters per second in the slowest fibers to over 100 meters per second in the fastest ones. The fast ones are wrapped in a fatty insulating sheath called myelin, which forces the signal to jump between gaps in the sheath, massively increasing the speed. Multiple sclerosis, by the way, is a disease where the immune system attacks myelin, degrading these insulating sheaths and disrupting neural signaling, which is why its symptoms can include numbness, weakness, vision problems, and difficulty with coordination. But here's what's really fascinating about neurons. When the electrical signal reaches the end of the axon, it usually doesn't jump directly to the next neuron. There's a tiny gap between neurons, called a synapse, typically about 20 nanometers wide, a distance so small that about 5,000 synapses would fit across the width of a human hair. When the electrical signal arrives at the synapse, it triggers the release of chemical molecules called neurotransmitters. These molecules float across the gap, land on receptors on the next neuron's dendrite, and either encourage or inhibit that neuron from firing its own electrical signal. So the nervous system uses a combination of electrical signals within neurons and chemical signals between neurons. It's an electrochemical communication network of staggering complexity. And the neurotransmitters themselves, serotonin, dopamine, norepinephrine, acetylcholine, GABA, glutamate, and dozens of others, these are the chemical vocabulary of your nervous system. Dopamine is involved in reward, motivation, and movement. Serotonin helps regulate mood, sleep, and appetite. Acetylcholine triggers muscle contractions and plays a role in memory. The balance and interplay of these chemicals underlies everything, from your ability to pick up a coffee cup to your capacity for love, fear, creativity, and abstract thought. When that balance goes wrong, too little dopamine in certain brain circuits, for instance, the result can be Parkinson's disease, with its characteristic tremors and difficulty initiating movement. Too much dopamine in other circuits is associated with symptoms of schizophrenia. Now, the brain itself. Your brain is, without exaggeration, the most complex object we know of in the universe. 3 pounds of tissue, about the consistency of soft tofu, folded and wrinkled to pack as much surface area as possible into the limited space of your skull. If you smoothed out all the folds of the cerebral cortex, the brain's outer layer, the part responsible for your most sophisticated mental abilities, it would cover roughly 2.5 square feet, about the size of a large pillowcase. All that surface area is crammed into a space the size of a coconut, and different regions of the brain handle different things. The occipital lobe, at the back of your head, processes vision. The temporal lobes, behind your temples, handle hearing and are deeply involved in memory and language comprehension. The parietal lobes, at the top, integrate sensory information and help with spatial awareness. The frontal lobes, behind your forehead, the largest lobes, and the most recently evolved, handle planning, decision-making, impulse control, personality, and working memory. And deep inside the brain, below the cortex, there are older structures: the amygdala, which processes fear and emotion; the hippocampus, which is essential for forming new memories; the thalamus, which acts as a relay station for sensory information; the hypothalamus, which regulates temperature, hunger, thirst, and the hormonal system; the cerebellum, at the back and below, which coordinates movement and balance with exquisite precision. And the way these regions connect to each other is equally remarkable. The brain's white matter, the tissue beneath the cortex, consists of billions of myelinated axons running in organized bundles, connecting distant brain regions the way highways connect cities. The corpus callosum, a thick band of about 200 million nerve fibers, connects the left and right hemispheres, allowing them to share information. When this connection is severed, as it sometimes is in a surgical procedure to treat severe epilepsy, the result is a split-brain patient, and the experiments done on these patients by Roger Sperry and Michael Gazzaniga in the 1960s and 1970s revealed some of the most fascinating things we know about how the two hemispheres work. The left hemisphere, in most people, dominates language production. The right hemisphere excels at spatial tasks and recognizing faces. In split-brain patients, information presented to one hemisphere literally can't reach the other, leading to bizarre situations where one hand might reach for an object that the other hand pushes away, because each hemisphere is making independent decisions. Sperry won the Nobel Prize for this work in 1981. One of the most dramatic demonstrations of how specific brain regions relate to specific functions came from an accident in 1848. A railroad construction foreman named Phineas Gage was working near Cavendish, Vermont, using a tamping iron to pack explosive powder into a hole drilled in rock. Something went wrong. The powder ignited prematurely, and the tamping iron, 3 feet 7 inches long and 1 1/4 inches in diameter, weighing over 13 pounds, shot upward through Gage's left cheek, behind his left eye, through the frontal lobe of his brain, and out through the top of his skull. It landed 80 feet away, covered in blood and brain matter, and Phineas Gage survived. Not only survived, he was reportedly speaking within minutes of the accident. He walked, with assistance, to an ox cart that took him to a doctor. He sat upright on the cart. The doctor, John Martin Harlow, later reported that Gage was alert and coherent, and that on the ride to the doctor's office, Gage casually told the men accompanying him about the accident, as though describing something mildly interesting that had happened to someone else. An iron rod had passed completely through his brain, and he was chatting about it on an ox cart. He lived for another 12 years, dying in 1860 from epileptic seizures that were almost certainly related to his injury. But he was changed. Before the accident, Gage was described as capable, reliable, and well-liked by his employers and crew. After the accident, according to Harlow's accounts, he became impulsive, irreverent, profane, and unreliable. He couldn't stick to plans. He made poor decisions. He was, in the words Harlow used, no longer Gage. His friends and acquaintances said the balance between his intellectual faculties and his animal propensities had been destroyed. Now, the details of Gage's personality changes have been debated by historians and neuroscientists. Some of Harlow's descriptions were written years after the fact, and they may be exaggerated. But the core finding has been supported by over a century and a half of subsequent research. The frontal lobes, particularly the prefrontal cortex, are critical for what we might call the executive functions of the mind: planning, decision-making, social behavior, impulse control, the things that make you you in a social and moral sense. Damage there can leave intelligence and memory intact while fundamentally altering personality and judgment, which tells you something profound and slightly unsettling about the relationship between your brain and yourself. The 20th century brought an explosion of new tools for studying the brain. Electroencephalography, EEG, allowed researchers to record the brain's electrical activity from electrodes placed on the scalp, revealing that the brain produces rhythmic waves of activity, different frequencies associated with different states of consciousness. Alpha waves when you're relaxed and awake, beta waves when you're focused and alert, theta waves during light sleep and meditation, delta waves during deep sleep. Right now, as you lie here, your brain is probably transitioning from alpha and beta waves toward the slower theta rhythms, your neural activity settling into the gentle patterns of drowsiness. And if you drift off, the delta waves will come, slow and deep, and your brain will enter the restorative phases of sleep that consolidate memories, repair tissues, and clear away the metabolic waste products that accumulate during waking hours. And then came functional brain imaging, PET scans, and especially fMRI, functional magnetic resonance imaging, which allowed scientists to watch the living brain at work in real time. Not its electrical activity, but its blood flow. Regions of the brain that are more active need more oxygen and glucose, so more blood flows to them, and fMRI detects these changes in blood flow. For the first time, you could ask a person to think about a word, or look at a face, or recall a memory, and see which parts of their brain lit up. It was like watching a city from above at night and seeing which neighborhoods had their lights on. But for all these tools and all these discoveries, the deepest questions about the brain remain open. How does the activity of 86 billion neurons, communicating through trillions of synapses, give rise to the experience of consciousness? How does electrochemical signaling become the feeling of seeing red, or tasting chocolate, or being in love? This is what philosophers call the hard problem of consciousness, and neuroscience hasn't solved it. We can map which brain regions are active during different experiences. We can identify neural correlates of consciousness, but we cannot yet explain how physical processes in the brain produce the subjective experience of being you. It's one of the great remaining mysteries of science, and it's happening right now, in the 3 pounds of tissue behind your forehead, as you listen to these words and wonder about it. But let's move on, because there's another system in your body that's equally astonishing and far less appreciated, a system that's protecting you right now from a world that is, frankly, trying to kill you: your immune system. And the story of how we came to understand it begins with one of the most terrifying diseases in human history, and an English country doctor who noticed something curious about milkmaids: smallpox. For centuries, it was one of the most feared diseases on Earth. Caused by the variola virus, it killed roughly 3 out of every 10 people it infected, and those who survived were often left with disfiguring scars, blindness, or other permanent damage. It's estimated to have killed over 300 million people in the 20th century alone, before it was finally eradicated. And the eradication of smallpox, the complete, total elimination of a human disease from the face of the earth, is arguably the greatest achievement in the history of public health, and it started with a milkmaid's hands. Edward Jenner was a country doctor in Berkeley, Gloucestershire, in the late 18th century. He'd been trained in London under the famous surgeon John Hunter, but he preferred rural life and returned to Gloucestershire to practice. And there was a piece of folk knowledge circulating among the dairying communities of rural England that caught his attention. Milkmaids who had caught cowpox, a mild disease that produced sores on the hands but was rarely dangerous, seemed to be immune to smallpox. They went through smallpox epidemics untouched while their neighbors sickened and died. Jenner decided to test this. On May 14, 1796, he took material from a cowpox sore on the hand of a milkmaid named Sarah Nelms and scratched it into the arm of an 8-year-old boy named James Phipps. The boy developed a mild cowpox infection and recovered quickly. Then, 6 weeks later, Jenner inoculated Phipps with actual smallpox material. And nothing happened. The boy didn't get sick. He was immune. This experiment, by modern standards, was ethically horrifying. Deliberately infecting a child with a potentially fatal disease to test a hypothesis. But in the context of the late 18th century, when smallpox was killing tens of thousands of people every year, and an earlier, more dangerous form of inoculation called variolation was already widely practiced, Jenner's gamble seemed less reckless than it does now. And it worked. Jenner published his results in 1798, coined the term vaccination from vacca, the Latin word for cow, and within a few years, vaccination against smallpox was spreading across Europe and eventually the world. Napoleon had his entire army vaccinated. Thomas Jefferson championed it in the United States. By the middle of the 19th century, vaccination was one of the most widely practiced medical interventions on Earth, and in 1980, after a decades-long global campaign led by the World Health Organization involving millions of health workers in some of the most remote regions on the planet, smallpox was officially declared eradicated. The last natural case occurred in Somalia in 1977. It remains the only human infectious disease ever to be completely eliminated, a disease that had killed hundreds of millions of people over thousands of years, gone forever, because an English country doctor paid attention to milkmaids. But here's what's fascinating. Jenner didn't know why vaccination worked. He didn't know about viruses. The smallpox virus wouldn't be visualized until the electron microscope was developed in the 1930s. He didn't know about the immune system in any modern sense. He just knew that exposure to cowpox protected against smallpox, and he had the courage and the scientific instinct to test it systematically. The mechanism, the actual biological explanation for why his vaccine worked, would take another century and more to unravel. Pasteur took the principle further. In the 1870s and 1880s, he developed vaccines against anthrax and rabies, this time understanding more about the microbial basis of these diseases. His public demonstration of the anthrax vaccine in 1881 at Pouilly-le-Fort, where vaccinated sheep survived exposure to anthrax while unvaccinated ones died, was a sensation, cementing both his reputation and the principle of vaccination in public consciousness. His rabies vaccine, developed using dried spinal cord material from infected rabbits, was first tested on a human in 1885, a 9-year-old boy named Joseph Meister, who had been badly bitten by a rabid dog and would otherwise have faced an agonizing death. The treatment worked. Meister survived, and Pasteur, who was not a physician and had no legal right to treat a patient, had taken an enormous personal and professional risk. Had the boy died, Pasteur could have faced criminal prosecution, but the vaccine worked, and Meister lived to old age. And that mechanism behind vaccination, the biological explanation for why exposing someone to a weakened or killed pathogen protects them against the real thing, is one of the most extraordinary stories in all of biology. Your immune system is, in effect, a military organization of breathtaking sophistication. It has scouts, soldiers, intelligence gatherers, memory banks, and weapons factories. It can identify and destroy invaders it has never encountered before, learn from the encounter, and remember the invader for decades, so that if it shows up again, the response is faster and more powerful. The first line of defense is simple and ancient. Your skin is a barrier, a wall that keeps most pathogens out. Your mucous membranes trap particles. Your stomach acid kills many organisms that you swallow. Tears and saliva contain enzymes that break down bacterial cell walls. These are the innate defenses, the ones you're born with, the ones that don't need to learn anything. But when a pathogen gets past these barriers, when bacteria enter through a cut, or a virus slips through your respiratory lining, that's when the adaptive immune system kicks in, and this is where things get truly remarkable. Your body has specialized white blood cells called lymphocytes, and the two main types are B cells and T cells. B cells produce antibodies, Y-shaped proteins that are custom designed to lock onto a specific invader. Each B cell produces antibodies that fit one particular molecular shape, called an antigen, on the surface of one particular pathogen. And here's the wild part. Your body doesn't wait until it encounters a pathogen to start making B cells. You are born with billions of B cells, each one producing a slightly different antibody, covering an almost unimaginable range of possible shapes. Your immune system has, in a sense, pre-manufactured locks for doors it has never seen. It's prepared for invaders that may not even exist yet. It's prepared for synthetic chemicals that have never existed in nature, for viruses that haven't evolved yet, for threats that are entirely hypothetical. The diversity is generated through a process of random gene rearrangement in your bone marrow, shuffling and combining gene segments to produce antibodies of enormous variety. It's like having a library with books written in every possible language, including languages that haven't been invented. When a B cell encounters a pathogen whose antigen matches its antibody, it activates. It multiplies rapidly, producing thousands of copies of itself, all churning out antibodies that flood the bloodstream and tag the invader for destruction. Other immune cells, macrophages, neutrophils, natural killer cells, find the tagged invaders and destroy them. And here's the key to vaccination. After the infection is cleared, some of those activated B cells don't die off. They become memory cells, long-lived sentinels that can persist in your body for years, even decades. If the same pathogen ever shows up again, the memory cells recognize it instantly and mount a response that's faster and far more powerful than the first one. The invader is wiped out before you even feel sick. That's what Jenner's vaccine did. The cowpox virus was similar enough to the smallpox virus that the immune response against cowpox, the antibodies, the memory cells, also worked against smallpox. The body was tricked into building defenses against smallpox by being exposed to something much less dangerous. And every vaccine since then, against polio, measles, tetanus, influenza, COVID-19, all of them, works on this same basic principle. You expose the immune system to something harmless that resembles the real threat, and the immune system builds its memory, its army, its arsenal, so that when the real threat arrives, it's ready. T cells, the other major type of lymphocyte, deserve their own moment, because they do something different and equally important. Some T cells, called helper T cells, coordinate the immune response, sending chemical signals that activate B cells and other immune components. Others, called cytotoxic T cells, or killer T cells, directly attack and destroy cells that have been infected by viruses. They don't just tag invaders the way antibodies do. They walk up to an infected cell and execute it, punching holes in its membrane and triggering it to self-destruct. This is crucial because viruses reproduce by hijacking your own cells, hiding inside them, and using their machinery to make more viruses. Antibodies can't reach a virus once it's inside a cell. Killer T cells can. They identify the infected cell by recognizing viral fragments displayed on its surface, and they eliminate it before it can produce more virus. And the whole system has built-in checks and balances to prevent it from attacking your own healthy cells. T cells are educated in the thymus, a small organ behind your breastbone, most active in childhood, where they're tested against samples of your body's own proteins. Any T cell that reacts too strongly to your own tissues is destroyed before it leaves the thymus. It's a quality control process that eliminates most self-reactive cells. Most, but not all. When this system fails, when immune cells do attack the body's own tissues, the result is autoimmune disease, conditions like type 1 diabetes, where the immune system destroys the insulin-producing cells of the pancreas, or rheumatoid arthritis, where it attacks the joints, or lupus, where it can target almost any organ. These are diseases of friendly fire, the immune system's awesome power turned inward. Allergies, too, are immune system misfires. When you're allergic to pollen or peanuts or cat dander, your immune system is treating a harmless substance as if it were a dangerous pathogen. It produces a type of antibody called IgE, which triggers the release of histamine and other chemicals that cause the symptoms you know: sneezing, itching, swelling, and in severe cases, anaphylaxis. Your immune system isn't broken. It's overreacting, mounting a full military response against something that poses no actual threat. It's like calling in an airstrike because someone knocked on your front door. There's so much more to say about immunity, about how cancer evades the immune system, about the promising field of immunotherapy that harnesses the immune system to fight tumors, about the microbiome and how the trillions of bacteria living in your gut interact with your immune cells, and we'll circle back to some of that. But first, let's go somewhere deeper. Let's go to the most fundamental level of your body's organization, to the molecule that contains the instructions for building and running every cell, every tissue, every organ, every system we've talked about tonight. Let's talk about DNA. The story of DNA is one of the great detective stories in the history of science, and it involves a cast of characters that includes a quiet monk with a garden full of peas, a brilliant woman whose contribution was stolen, two ambitious young scientists who claimed most of the credit, and a molecule so elegant that when its structure was finally revealed, one of the discoverers reportedly walked into a pub in Cambridge and announced that they had found the secret of life. Let's start with the monk. Gregor Mendel was an Augustinian friar in Brno, in what is now the Czech Republic, and in the 1850s and 1860s, he conducted a series of experiments that would eventually become the foundation of modern genetics. He didn't work with DNA. Nobody knew DNA existed yet. He worked with pea plants, thousands of them. He crossbred peas with different traits, tall plants with short plants, purple flowers with white flowers, smooth peas with wrinkled peas, and carefully recorded the ratios of traits that appeared in the offspring. And what he found was remarkable. Traits didn't blend. A cross between a tall plant and a short plant didn't produce a medium-sized plant. It produced tall plants. But when those tall plants were crossed with each other, some of their offspring were short. The shortness hadn't disappeared. It had been hidden, carried silently for a generation, and then reappeared. Mendel worked out the mathematical ratios, 3:1 for the second generation, and proposed that traits are controlled by discrete factors, now called genes, that come in pairs, one from each parent. Some factors are dominant, expressing themselves whenever present. Others are recessive, expressing themselves only when no dominant factor is around to override them. Mendel published his work in 1866 in the Proceedings of the Natural History Society of Brno, and essentially nobody noticed. The paper was sent to about 40 libraries and scientific institutions. It was cited a handful of times over the next 34 years, mostly in passing. Part of the problem was that Mendel was a monk in a provincial city, not a professor at a major university. Part of it was that his mathematical approach, ratios, probabilities, statistical analysis of breeding results, was unfamiliar to the biologists of his time who weren't used to thinking about biology in mathematical terms. And part of it was simple bad luck. The right people didn't read it, and the wrong people ignored it. Mendel died in 1884, at 61, having become more consumed by administrative duties as abbot of his monastery than by scientific research. He reportedly told a colleague, "My time will come." He was right, but he never saw it. It wasn't until 1900 that 3 other scientists, working independently, Hugo de Vries in the Netherlands, Karl Correns in Germany, and Erich von Tschermak in Austria, each rediscovered Mendel's principles through their own breeding experiments, and then found his original paper, giving him posthumous credit. The science of genetics was born, or rather reborn, and the race was on to find the physical substance that carried these hereditary factors. That substance turned out to be deoxyribonucleic acid, DNA, though it took decades to prove it. DNA was first isolated in 1869 by a Swiss chemist named Friedrich Miescher, who extracted a phosphorus-rich substance from the nuclei of white blood cells found in pus-soaked bandages. He called it nuclein, but for a long time, most scientists thought that proteins, not nucleic acids, were the carriers of genetic information. Proteins were complex, diverse, capable of enormous structural variety. DNA seemed too simple, just 4 chemical building blocks, called nucleotides, repeated over and over. How could something that repetitive encode the instructions for building an entire organism? The proof that DNA was indeed the genetic material came in stages. In 1944, Oswald Avery, Colin MacLeod, and Maclyn McCarty at Rockefeller University showed that DNA from one strain of bacteria could transform another strain, transferring heritable traits. In 1952, Alfred Hershey and Martha Chase used bacteriophages, viruses that infect bacteria, to demonstrate that it was DNA, not protein, that entered the bacterial cell and directed the production of new viruses. The evidence was mounting, but the critical question remained: what is the structure of DNA? How is this molecule arranged in 3-dimensional space? And how does that structure allow it to carry and copy information? This is where the story gets personal, and complicated, and not entirely comfortable. By the early 1950s, several groups were racing to determine the structure of DNA. At King's College London, a physicist named Rosalind Franklin was using a technique called X-ray crystallography to produce images of DNA fibers. Crystallography works by firing X-rays at a crystalline substance and analyzing the pattern of diffracted rays to determine the arrangement of atoms. It's painstaking, technical work that requires enormous skill. And Franklin was extraordinarily skilled. In May 1952, she and her graduate student Raymond Gosling produced an image of DNA, known as Photograph 51, that was stunningly clear. The X-shaped pattern in the image was a signature of a helical structure. Franklin was cautious, methodical. She was working toward a definitive analysis, not rushing to publish. Meanwhile, at Cambridge, James Watson and Francis Crick were building physical models of possible DNA structures, trying to find an arrangement that matched all the known chemical and physical data. Watson was an American biologist, brash and ambitious, just 24 years old, with a mop of unruly hair and an almost irritating level of confidence. Crick was a British physicist in his mid-30s, talkative, enthusiastic, and brilliant at synthesizing information from different sources. His colleagues at Cambridge joked that Crick's voice could be heard from any point in the Cavendish laboratory. Together, they were an extraordinary team. Watson brought the biological intuition, Crick brought the physics and the mathematical rigor, and both brought a fierce, competitive drive. They knew they were in a race, not just with Franklin and Wilkins at King's College, but with Linus Pauling at Caltech, one of the most celebrated chemists in the world, who had already solved the structure of proteins and was now turning his attention to DNA. And in January 1953, without Franklin's knowledge or consent, her colleague Maurice Wilkins showed Watson photograph 51. Watson later wrote that the moment he saw it, his mouth fell open and his pulse began to race. The helical structure was unmistakable. Armed with this crucial data Franklin had produced through her own meticulous work, Watson and Crick built their model. On February 28, 1953, they arrived at the structure that would become one of the most famous scientific discoveries of all time, the double helix. Two strands of DNA, wound around each other in a spiral, connected by pairs of nucleotide bases. Adenine always pairs with thymine. Guanine always pairs with cytosine. The base pairing is the key to everything, because it means that each strand of the double helix contains all the information needed to reconstruct the other. If you pull the strands apart, each one serves as a template for building its partner. That's how DNA replicates, how your cells copy their genetic information every time they divide. Watson and Crick published their paper in the journal Nature on April 25, 1953. It was a single page long, and in one of the most famous understatements in scientific literature, they wrote, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." They knew exactly what they had. Watson, Crick, and Wilkins shared the Nobel Prize in 1962. Franklin did not. She had died of ovarian cancer in 1958, at the age of 37, and the Nobel Prize is not awarded posthumously. The question of how much credit she deserved, and how much was denied her, has been debated ever since. Most modern historians of science consider her contribution essential and under-recognized. But let's step back from the human drama for a moment and think about what this molecule actually does, because it's staggering. Your DNA contains about 3 billion base pairs, 3 billion rungs on that spiral ladder, packed into the nucleus of nearly every cell in your body. If you stretched out all the DNA in a single cell, it would be about 2 meters long. If you stretched out all the DNA in your entire body, all 37 trillion cells worth, the total length would be roughly twice the diameter of the solar system. All of it packed into a body that fits in a bed. And that DNA is an instruction manual. It encodes the sequences of amino acids that make up every protein your body produces. Proteins are the workhorses of biology. They're the enzymes that catalyze chemical reactions, the structural components that give cells their shape, the antibodies that fight infections, the hemoglobin that carries oxygen, the receptors that receive signals, the motors that contract muscles. Your body uses roughly 20,000 genes to produce a vastly larger number of different proteins through various splicing and modification mechanisms, and those proteins, in their staggering variety, build and run your body. The process goes like this. When a cell needs a particular protein, the relevant stretch of DNA, the gene, is copied into a single-stranded molecule called messenger RNA. This RNA travels out of the nucleus to structures called ribosomes, which read the RNA sequence 3 letters at a time. Each group of 3 letters Each codon specifies one particular amino acid. Transfer RNA molecules carry the appropriate amino acids to the ribosome, which links them together, one by one, into a growing protein chain. When the chain is complete, it folds into a specific three-dimensional shape that determines its function. This flow of information, from DNA to RNA to protein, is so fundamental to biology that Francis Crick called it the central dogma of molecular biology, and the ability to read and manipulate this code has transformed medicine. The Human Genome Project, launched in 1990 and completed in 2003, sequenced the entire human genome, all 3 billion base pairs. It was one of the most ambitious scientific endeavors in history, involving laboratories in 6 countries and costing roughly $3 billion. Today, you can have your genome sequenced for a few $100s in a matter of days. That collapse in cost and time has opened up the era of genomic medicine, where doctors can identify genetic mutations that cause disease, tailor treatments to an individual's genetic profile, and screen for inherited conditions before symptoms appear. Think about what that means for medicine. If you know that a particular patient has a mutation in a gene called BRCA1 that dramatically increases their risk of breast and ovarian cancer, you can screen for that mutation, monitor more closely, and consider preventive measures. If you know that a patient's tumor has a specific genetic signature, you can choose treatments that target that signature rather than using a one-size-fits-all approach. This is precision medicine, and it's already transforming oncology, cardiology, and rare disease treatment. And then came CRISPR. In 2012, Jennifer Doudna at UC Berkeley and Emmanuel Charpentier, then at Umeå University in Sweden, published a paper describing a tool that could edit DNA with unprecedented precision. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is actually a defense mechanism that bacteria use against viruses, but Doudna and Charpentier realized it could be adapted into a molecular scalpel capable of cutting DNA at any desired location and inserting, deleting, or replacing specific sequences. It's like having a find and replace function for the genetic code. They shared the Nobel Prize in Chemistry in 2020, and the implications of their discovery are still unfolding. CRISPR has already been used to treat sickle cell disease in clinical trials, with remarkable early results. The potential for treating genetic diseases, engineering more resilient crops, and even modifying the genes of future generations is enormous. And so are the ethical questions that come with it. But here's what I find most remarkable about all of this. The molecule that carries the instructions for building you, DNA, is essentially the same molecule that carries the instructions for building a fruit fly, a whale, a redwood tree, a bacterium, a mushroom. The genetic code is nearly universal across all life on Earth. The same codons that specify amino acids in your cells specify the same amino acids in the cells of every other organism. This isn't a coincidence. It's evidence of common ancestry. All life on Earth, every species that has ever lived, descended from a single common ancestor, a single cell, billions of years ago. The DNA in your cells is a direct descendant of the DNA in that first cell, modified and elaborated over 4 billion years of evolution, but fundamentally the same code, the same alphabet, the same operating system. And this leads us to something that might be the most surprising discovery about the human body in recent decades. Because for a long time, we thought of the body as, well, ours, a human organism. But it turns out we're not alone in here, not even close. Your body is home to roughly 38 trillion microorganisms, bacteria, fungi, viruses, archaea, living on your skin, in your mouth, in your gut, in your respiratory tract, in places you've never thought about. The number of microbial cells in and on your body is roughly equal to the number of your own human cells. By some estimates, the microbial genes in your body outnumber your human genes by a factor of 100 to 1. You are, in a very real sense, an ecosystem, a walking coral reef, a planet with inhabitants. And these microbes aren't freeloaders. Most of them are doing things you need. The bacteria in your gut microbiome, which contains several hundred to over 1,000 different species, depending on the individual, help you digest food that your own enzymes can't break down, particularly complex carbohydrates and plant fibers. They produce vitamins your body can't make on its own, including vitamin K, which is essential for blood clotting, and several B vitamins. They train your immune system during infancy and early childhood, teaching it to distinguish friend from foe, which is one reason why children raised in extremely sterile environments sometimes develop more allergies and autoimmune conditions, because their immune systems never got the microbial education they needed. The gut bacteria protect you against pathogenic bacteria by occupying the ecological niches those pathogens would otherwise exploit, competing for resources and producing antimicrobial compounds. And perhaps most surprisingly, they produce neurotransmitters, including serotonin, of which roughly 90% of the body's supply is actually produced in the gut, not the brain, that influence your mood, your sleep, and your behavior through what scientists call the gut-brain axis, a bidirectional communication highway between your digestive system and your central nervous system. There's growing evidence that the composition of your gut microbiome affects everything from your weight to your mental health to your susceptibility to autoimmune diseases. This is a genuinely new frontier in medicine. The Human Microbiome Project, launched in 2007, has been mapping the microbial communities that inhabit different parts of the body, and the findings are reshaping how we think about health and disease. Conditions that seem to have nothing to do with bacteria, obesity, depression, inflammatory bowel disease, even some neurological disorders, may be influenced, in ways we're only beginning to understand, by the composition of our microbiomes. Fecal microbiota transplantation, which is exactly what it sounds like, has proven remarkably effective in treating recurrent Clostridioides difficile infections, where a patient's own gut microbiome has been devastated by antibiotics and can't recover on its own. You repopulate it with a healthy person's microbiome, and the infection clears. It's crude, it's inelegant, and it works. And this brings us full circle, in a way, because the recognition that the human body is a community, a collaboration between human cells and microbial cells, between our genome and the genomes of our microscopic tenants, echoes something that's been true at every level of the body's organization. Your body is not a machine in any simple sense. It's not a collection of independent parts that can be understood in isolation. It's a system, a network, an ecology, where everything influences everything else. Your heart rate affects your brain function. Your gut bacteria influence your immune system. Your immune system shapes the development of your brain. Your hormones, produced by glands scattered throughout your body, carry chemical messages that coordinate growth, metabolism, reproduction, stress responses, and circadian rhythms. Your endocrine system, your nervous system, and your immune system aren't separate systems. They're deeply interconnected, constantly talking to each other through a web of chemical signals so complex that we're still mapping it. Consider your lungs, for instance. Each lung contains about 300 million tiny air sacs called alveoli, with a total surface area roughly the size of a tennis court. That enormous surface area, folded and packed into your chest cavity, is where gas exchange happens. Oxygen molecules crossing from the air in the alveoli into the blood in the surrounding capillaries, and carbon dioxide crossing the other way. The membrane between air and blood is about half a micrometer thick, roughly 1/100th the width of a human hair. It has to be that thin for gases to diffuse across it efficiently, and you breathe about 20,000 times a day, drawing in about 11,000 liters of air, extracting the 21% of that air that's oxygen, and passing it into your blood for distribution to every cell in your body. Or consider your kidneys, a pair of bean-shaped organs in your lower back that most people never think about until something goes wrong with them. Each kidney contains about 1 million tiny filtering units called nephrons. Blood flows through these nephrons under pressure, and the nephrons filter out waste products, urea, creatinine, excess salts, toxins, while carefully reabsorbing the water, glucose, amino acids, and electrolytes your body needs. Your kidneys filter your entire blood supply roughly 40 times a day, processing about 180 liters of fluid, but producing only about 1.5 liters of urine, because they reabsorb more than 99% of the water they filter. The precision of this process is remarkable. Your kidneys regulate the concentration of sodium, potassium, calcium, and other ions in your blood within extraordinarily tight limits, because even small deviations can disrupt the electrical signaling in your heart and nervous system. They also regulate blood pressure, produce hormones that stimulate red blood cell production, and activate vitamin D for bone health. 2 organs, each about the size of your fist, performing critical chemistry around the clock. Or think about stress as an example of how interconnected everything is. When you perceive a threat, and it doesn't have to be a physical threat, it can be a looming deadline or a difficult conversation, your brain's hypothalamus signals your adrenal glands, sitting on top of your kidneys, to release cortisol and adrenaline. Your heart rate increases, your blood pressure rises, your muscles tense, your digestion slows down. Energy is being diverted from long-term maintenance to immediate survival. Your immune system shifts, boosting some defenses and suppressing others. All of this happens in seconds, coordinated across multiple organ systems simultaneously. And when the threat passes, the whole system dials back down, returning to baseline through a network of feedback loops that would make an engineer weep with admiration. And healing. Your body's ability to heal itself is something we take for granted, but it's genuinely extraordinary. When you cut your finger, what follows is an orchestrated sequence of events involving dozens of cell types, hundreds of signaling molecules, and multiple organ systems working in precise coordination. Platelets aggregate at the wound site, forming a clot. White blood cells arrive to fight potential infection. Inflammatory signals recruit more immune cells and increase blood flow to the area. Fibroblasts begin producing collagen, building new connective tissue. Epithelial cells at the wound's edges start dividing and migrating across the wound's surface, eventually closing it. New blood vessels grow into the healing tissue. Over days and weeks, the temporary repair is gradually remodeled into something close to the original tissue. Not always perfect. Scars are the evidence of repair that didn't quite match the original, but functional. Your body rebuilt itself. No one told it to. No one directed the process. Billions of cells, each responding to local chemical signals, collectively producing a coordinated result that looks for all the world like intelligent design. And bones are even more impressive. A broken bone, properly set, can heal to full strength. The process involves a cascade of cellular activity, blood clot formation at the fracture site, recruitment of stem cells from the bone marrow, gradual replacement of the clot with cartilage and then with woven bone, and finally remodeling of the woven bone into the dense, organized lamellar bone that gives the skeleton its strength. The whole process can take weeks to months, and when it's done, The bone at the fracture site is often stronger than it was before. Your skeleton is not a static scaffold. It's a living tissue, constantly being broken down and rebuilt by specialized cells, osteoclasts that dissolve old bone, and osteoblasts that build new bone, in a continuous cycle of renewal that remodels your entire skeleton roughly every 10 years. The skeleton you have now is not the skeleton you had a decade ago. The atoms are different. The structure has been rebuilt from scratch. But the blueprint, the information encoded in your DNA, interpreted by your cells, remains constant. And speaking of your skeleton, it's worth noting just how beautifully it's engineered. The femur, the thigh bone, is the longest and strongest bone in the body. It can support roughly 30 times your body weight before fracturing. The trabecular bone inside it, the spongy-looking internal structure, is arranged in a lattice of tiny struts and plates that follow the lines of mechanical stress, placing material exactly where it's needed and removing it where it's not. This pattern was discovered in the 1860s by the German anatomist Julius Wolff, who formulated what's now called Wolff's Law. Bone remodels itself in response to the forces placed upon it. Load a bone repeatedly and it gets stronger. Unload it, as happens to astronauts in microgravity, and it weakens. Your skeleton is not just a support structure. It's a responsive, adaptive, self-optimizing framework. And your digestive system, here's something to marvel at. The journey from mouth to exit is roughly 30 feet long in an adult, and the process of digestion involves an extraordinary coordination of mechanical, chemical, and biological action. Your stomach produces hydrochloric acid strong enough to dissolve metal, about 2 liters of it per day, and yet the stomach doesn't dissolve itself, because the cells lining it secrete a thick layer of protective mucus that's constantly being replaced. When that mucus layer fails, you get an ulcer. Your small intestine, where most nutrient absorption happens, has an inner surface covered with tiny finger-like projections called villi, and those villi are themselves covered with even tinier projections called microvilli. This fractal-like folding increases the absorptive surface area of your small intestine to roughly the size of a studio apartment, about 32 square meters, all packed into a tube about 20 feet long and an inch in diameter. Evolution solved the problem of maximizing surface area within a limited space by folding upon folding upon folding, and the result is an absorptive surface that can extract almost every usable molecule from the food you eat. Your liver can regenerate. If you lose up to 75% of your liver to surgery or injury, the remaining portion will grow back to nearly its original size within weeks. It's one of the only organs in the human body with this capacity, and it was actually known to the ancient Greeks. The myth of Prometheus, chained to a rock while an eagle eats his liver every day, only for it to regrow each night, is a weirdly accurate metaphor for hepatic regeneration. And your entire inner surface, the lining of your stomach, your intestines, your lungs, is constantly renewing itself. The cells lining your stomach are replaced every few days because they're constantly exposed to the harsh acid environment of gastric digestion. Your skin cells turn over roughly every 2 to 3 weeks. The red blood cells we talked about earlier last about 4 months before they're recycled. Your taste buds replace themselves every 10 days or so. You are, at a cellular level, not the same person you were a few years ago. Most of the atoms in your body have been replaced multiple times since you were born. You're a pattern, a process, more than you are a thing. The matter flows through you like water through a river. What persists is the pattern, the organization, the information encoded in your DNA and expressed through billions of cells cooperating in ways that still, after all these centuries of study, fill us with wonder. And that wonder, I think that's the right note to end on, because as you lie here tonight, breathing slowly, feeling the weight of your body settling into the mattress, there's a temptation to think that we've figured the body out, that the major discoveries are behind us, that modern medicine knows how everything works, and it's just a matter of fine-tuning. But that's the same mistake the physicists made at the end of the 19th century, when they thought physics was essentially complete. We know so much more than Hippocrates did, so much more than Galen, so much more than Vesalius and Harvey and Semmelweis and Pasteur. But the body keeps surprising us. We still don't fully understand consciousness. We don't know why we sleep. We know sleep is essential. We know terrible things happen without it. We know that even a few days without sleep can cause hallucinations, cognitive impairment, and eventually death. But the fundamental biological reason for sleep remains debated. Is it for memory consolidation? For clearing metabolic waste from the brain through the glymphatic system, a network of channels that opens up during sleep and flushes out proteins like amyloid beta that are associated with Alzheimer's disease? For tissue repair? For immune function? Probably all of these. But the core question, why did evolution produce an organism that must spend a third of its life unconscious and vulnerable, hasn't been definitively answered. We don't fully understand chronic pain, or the placebo effect, that remarkable phenomenon where a sugar pill, if the patient believes it's medicine, can produce real, measurable physiological changes, including the release of endogenous opioids in the brain. We don't know exactly why some people's immune systems attack their own bodies while others don't. We're only beginning to understand the microbiome and the ways in which our microbial partners influence our health and behavior. We don't know exactly how general anesthesia works. We use it every day, in every hospital in the world, and we understand a lot about which drugs affect which receptors. But the full mechanism by which consciousness is switched off and back on remains unclear. We don't fully understand aging, why cells senesce, why telomeres shorten, why organisms decline, why there seems to be a built-in limit to the human lifespan somewhere around 120 years, and whether that limit is fixed or flexible. Every answer reveals new questions. That's the nature of it, and that's fine, because the story of the human body, the story we've been walking through tonight, has never been about reaching a final answer. It's been about the asking. It's been about the looking. From Hippocrates, who said, "Disease has natural causes," to Vesalius, who opened the body and dared to say Galen was wrong, to Harvey, who measured and calculated and proved that blood circulates, to Leeuwenhoek, who saw a universe of tiny creatures in a drop of water, to Semmelweis, who knew the answer but couldn't make anyone listen, to Pasteur and Koch, who proved that germs cause disease, to Watson and Crick and Franklin, who revealed the structure of the molecule of life, to the thousands of researchers today who are mapping the brain, engineering the genome, exploring the microbiome, and pushing into territories that would have seemed like science fiction a generation ago. Every one of them was driven by the same impulse, the same one you might be feeling right now, even as sleep starts to pull at the edges of your awareness. Curiosity, the urge to know, what's in there? How does it work? Why does it do that? And the answer, the honest answer, is that it does what it does because billions of years of evolution have shaped it, cell by cell, mutation by mutation, into something that works astonishingly well, not perfectly. The body has its flaws. The recurrent laryngeal nerve that takes a bizarre detour down to the chest and back up to the throat. The narrow human birth canal. The blind spot in each eye where the optic nerve passes through the retina. The appendix that can suddenly try to kill you. These quirks and imperfections are the fingerprints of evolution, the evidence that the body wasn't designed from scratch by an engineer, but gradually modified over eons from structures that served different purposes in our ancestors. We share 98% of our DNA with chimpanzees, 60% with fruit flies, and about 50% with bananas. The code is the same. The variations are what make us different. And yet, within that shared code, the human body has evolved some extraordinary specializations. Our brains, relative to body size, are among the largest of any animal. Our hands, with their opposable thumbs and extraordinary dexterity, can thread a needle, play a piano, perform microsurgery. Our vocal apparatus, the larynx, the tongue, the lips, the soft palate working together, can produce the full range of human speech, an ability that no other species shares in anything like our complexity. Our upright posture, which freed our hands for tool use and carrying, came at a cost. Back pain, knee problems, a birth canal that's uncomfortably narrow for the size of our babies' heads, but it allowed us to travel vast distances efficiently and to see predators and prey across open grasslands. Every feature of your body is a compromise, a trade-off, a solution that evolution arrived at not by design, but by the accumulated weight of billions of small random variations, tested against the unforgiving filter of survival and reproduction. But here's the thing that gets me every time I think about it. You are the current endpoint of an unbroken chain of survival stretching back 4 billion years. Every one of your ancestors, going back to the first single-celled organism in the primordial ocean, survived long enough to reproduce. Every single one. Through ice ages and volcanic winters, through mass extinctions and pandemics, through predators and parasites and famines. Not one link in that chain was broken. The body you're lying in tonight is the product of that entire history. Refined, adapted, elaborated, layer upon layer, system upon system, from the simplest biochemistry to the staggering complexity of the brain you're using to understand these words. And right now, that body is doing what it does best. It's taking care of you. Your heart is beating. Your lungs are breathing. Your immune system is on watch. Your cells are dividing, repairing, communicating. Your brain is slowing its rhythms, easing from the alert alpha waves of wakefulness into the softer theta rhythms of drowsiness, and soon, if you let it, into the deep delta waves of restorative sleep. Trillions of cells, working together in a collaboration so seamless that you don't notice it happening, so reliable that you trust it with your life every single night. You don't have to understand it for it to work. You don't have to know about sinoatrial nodes and hemoglobin and neurotransmitters and killer T cells for your body to do its job. It was doing all of this long before Hippocrates, long before Vesalius, long before anyone thought to ask how. But knowing, even a little, adds something. A sense of awe, maybe. A quiet appreciation for the machinery that carries you through every day. The body is the most intimate thing you will ever have. The thing closest to you. The thing that is you. And it is, without exaggeration, the most complex and beautifully organized structure in the known universe. So let that settle. Let your breathing slow. Let the weight of the day ease out of your muscles, your shoulders, your jaw. The chemistry of sleep is already working. Melatonin rising, cortisol falling, your body temperature dropping slightly, your brain quieting. 37,000 billion chemical reactions, and not one of them needs your permission. You are held, tonight, by 4 billion years of practice. Rest in that. Sleep well.